Welcome back to Day 2 of Advances in Drug Discovery and Development 2023. I'm Elizabeth Bowen, Conference Producer at Technology Networks, and I'm here to introduce our next talk, Call to Action, Critical Need to Address the Paucity of Novel Oncology Targets. And I'm pleased to welcome Dr. Brian Lamon. Dr. Lamon joined Caris Life Sciences in September 2020 and currently serves as Senior Vice President, Chief Business Officer, and Head of Biopharma Business Development. He's responsible for developing the company's partnering and business development strategy and translating that strategy into actionable and achievable goals for the broader Caris organization. Dr. Lamon received his BA in Biology and French from Lafayette College and earned his PhD in Pharmacology from New York Medical. Prior to joining industry, he was Assistant Dean of Research Development and Assistant Professor, Department of Oncology and Laboratory Medicine at Weill Medical College of Cornell University in New York. A warm welcome to you, Dr. Lamon. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk. So, feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Dr. Lamon's presentation. You can also benefit from some fantastic supporting literature, which you can download for free below the video player. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. I will now hand over to Dr. Lamon to begin his presentation.
Thank you very much, Elizabeth, and absolutely questions and comments are very much welcome. So please post those during the presentation and then at the end for Q&A. So thank you for the opportunity to present on this topic today. I think it's very timely. It's a topic that has kind of emerged from the broader background of the industry and the market and some of the different pressures that influence how we think about discovery, frankly, how we think about everything from discovery through development and commercialization. Again, my name is Brian Lamon. My role at Caris Life Sciences is primarily to work with biotech and large pharma and everything in between to help advance the next wave of therapeutics. We do that at the early discovery phase, which is really the topic of the latter half of my talk today, but we also do that throughout development, first in human, getting to regulatory approval, commercialization, and lifecycle management. So the title here is, as you heard from Elizabeth, a call to action, and the background of that argument is listed here. And the hypothesis is that we have a paucity of truly novel targets in oncology, and this is something we have to address as a community. That means everyone listening. That means everyone in healthcare. Particularly those involved in discovery and development. This is something we've been talking about for quite some time at Caris, and you'll see a little bit about how that story has evolved over time. We've had a couple of years now of discussions on this topic with biotech and pharma, but this is really the first time we're presenting it a little more broadly and externally. So I'm genuinely excited to be able to present it, but also to hear the feedback and understand whether or not this resonates with different areas of the industry. So we'll step back and look a little bit about what's shaping the early discovery. And I want to start with a positive. You can see here lots to be very, very proud of in the industry over the last couple decades. The last 2 decades in particular have been prominent with lots of advances, innovation. You can measure that innovation and those breakthroughs in many different ways. A couple different things are shown here. On the top left, you can see the cumulative number of approvals over the years, and then below that on the left-hand side, you can see the rate of approvals. Both are going up pretty strikingly. This means more opportunities for patients to live longer. This means better outcomes for patients. In fact, if you look back in the mid to late 2010s and 2016 to 17, the most dramatic reduction in overall cancer mortality occurred in that time period. And from 2016 to 2017, over 2% of a reduction, which is pretty striking. Believed to be largely driven by advances in immuno-oncology, advances in targeted therapies, the number of patients, particularly in non-small cell lung cancer, which have benefited from those therapeutics, spurned a lot of these positive changes in mortality. However, you can see on the right-hand side, it's not only in lung cancer, it's really across the board. You can see across different tumor types from top to bottom. You can see some of the different tumor types that have had approvals from 1 all the way to 10 or beyond. Most of the tumors on here have had significant innovation and have had breakthroughs. You have tumor types like melanoma, which have been completely transformed by immunotherapy, talking about a cure in a disease that was once, a death sentence for almost everyone. However, you still have, brain cancer, you have pancreatic cancer, and others where advances have not been made. But overall, I think the major takeaway is that really encouraging progress across the cancer treatment landscape. So what are some of the other things that have emerged with all this innovation and all these positive outcomes for patients? Well, we've seen a really interesting trend, and this is in parallel with all that innovation. We've seen a significant increase in the herding of biopharma pipelines around a small set of targets. And you can see that here in this figure in a couple different ways. If you look at all therapeutics, on the y-axis, you have the number of assets, so the number of therapies per target. On the x-axis, you have time. You can see over the last 20 years or so. You can see by herding, what we mean is there's a concentration of pipelines around the same targets, right? Less and less diversity of targets. So for example, in 2000, on the left-hand side, there were on average 3 assets per actively investigated target. This is more than doubled across all therapies, reaching 7 assets per target in 2022. Even more striking and highly relevant to today's talk is in oncology. You can see in purple there, in light blue, you can see in oncology, the number of assets in clinical development is rapidly outpacing the number of biological approaches. So we've gone from less than 2 assets per target to 5 times that amount, almost 10 from 2000 to 2022. So what does that mean? That means that when we say target, we think about biological approach. We're essentially thinking about how do we kill a tumor, right? How do we attack a cancer? How do we reduce the number of metastases, et cetera? That may be an oncogene, that may be a homing beacon on the cell surface. We want to rid the patient of that tumor, and you're seeing more and more companies go after the same target. So I want to be as objective as I can throughout this presentation, and it's not to say that herding is only a bad thing. However, there are some things to acknowledge that things like herding does drive benefit in different ways. For example, herding has led to an increase in urgency in the marketplace, right? Companies are highly incentivized to develop a truly best-in-class asset against a given target. So it may not be the first-in-class medicine that gets through that is the most effective. It creates competition in the marketplace. It brings new opportunities and new options and new therapeutic options to patients. But it also, again, starts to beg the question as to what is the cost of this? Is there an opportunity cost here that we should be aware of? So if you dive a little deeper and you look at what is driving the increase in herding has been a broad trend really across pipelines, but also has been most prominent. You can see that on the left-hand side across the most frequently pursued targets. So for example, the top 5 most active targets in a given year have had a 5-fold increase in the assets per target between 2000 and 2020. You're all aware of the usual suspects here. You can see those on the right-hand side. In 2022, we have 60 assets in clinical development against CD19. We have 56 assets in clinical development against PD-1 or PD-L1, almost 40 assets in clinical development targeting HER2. So what are the questions that continue to arise there? We want to challenge the paradigm and say, is there really a clinical need? Is there really a viable commercial path for 60 CD19s? Is there really a need to have a 9th, a 10th, or a 50th PD-1 or PD-L1? And is that the best use of pharma and biotech R&D resources and investments? Again, to play a little bit of devil's advocate here, it's not that cut and dry. It would be nice to say, let's go be innovative all the time, but that's not always practical. And you can acknowledge other strategic reasons that companies may pursue Me Too's. The monumental and transformative impact that IO has had on cancer has been incredible. It's impacted not only patients and outcomes, but impacted the industry at large. On the left-hand side, you can see it's a little bit small font, but on the y-axis, you basically have from top to bottom the years from 2014 through 2023. And along the top, you have different tumor types. And each of those little colored dots in that figure represent an approval in a given indication by a PD-1 or PD-L1. And that's why we have 60 plus indications and growing in this class. And yes, this is having transformative impact on patients. And there's a reason that many companies want to go in and explore opportunities with PD-1s and PD-L1s, even if they're not first, second, or fifth to market. I won't go through all of them, but a few examples would be PD-1 is a backbone, right? There was a period when if you wanted to do combination therapy, and in many cases you had to do so, you had to have a PD-1 backbone, whether you had a novel IO mechanism or whether you had a non-IO mechanism. The Mercks, the BMSs of the world that were first in this space were basically controlling a lot of the PD-1 ability to do combinations. So those companies, those other companies with those novel assets would have to go out and buy PD-1s if there was not some sort of collaboration with some of the initial big pharma. The price of these medicines obviously can be over $100,000 or more a year. So this can be very expensive. That's just for one patient. Imagine running a trial and you need that PD-1 as a combination therapy, or you need a control arm that has a PD-1 where you're going to have to be funding that yourself. And that can be cost prohibitive. It certainly wasn't the case for the early PD-1s, which were usually going against chemotherapy. Chemotherapy regimens would be pennies on the dollar or less, frankly, from a cost perspective. So you had companies like GSK, like Merck Serono, Regeneron, et cetera, come to market with novel PD-1s, PD-L1s, and that opened up tremendous combination potential for their portfolios. Another important factor is all PD-1s and PD-L1s are not created equal. We have seen much data in indications or tumors like bladder cancer, renal cell carcinoma, where one asset, one therapy may succeed and have a positive study and another may fail. We won't get into the differences there, but that's certainly a legitimate argument why we need more innovation in the checkpoint space. But it is inevitably good for patients to have more and more opportunities to receive these medicines if there's a good chance that they're going to respond and be able to combat their cancer. So nevertheless, almost 60 different PD-1s and PD-L1s is a large number. So who's driving this, right? Why is this happening? What are the pressures in the industry that's driving this herding of these assets? And if you press a little deeper, you can see that the degree of herding around targets that are pursued by top 10 pharma and probably a little bit larger than that, the top 20, top 30, but shown here are the top 10 pharma, there's significant herding in the big pharma pipelines. And for the purposes of this slide, define significant herding as more than 5 assets per target. You can see here that in the year 2000, we had 16% of assets from top 10 pharma companies were targets that exhibited significant herding versus today, or versus the last couple of years, that number is almost 70%. So big pharma is clearly leading the way here, not only in absolute numbers, but also in percentages and the balance of their pipelines, of their portfolios, but they're not the only ones. And I think that rate that we saw with top pharma also is exhibited in smaller companies, in the mid caps and biotechs. And I think it's because there's an ecosystem impact here. Pharma has had, major patent cliffs. We've had something like an immunotherapy, a PD-1 comes along. You're looking at a Merck pembrolizumab, which I believe will be north of $90 billion in aggregate by the end of this year. These aren't, blockbusters. These are mega blockbusters. So it's been transformative for patients, but it's also creating high expectations and potential gaps in revenue. So now we have pharma companies looking at, what are we going to do next? What's next? What's going to replace an Opdivo? What's going to replace a Keytruda, nivolumab, or pembrolizumab? And that's a daunting task for leadership in these companies. So pharma has to replace and provide answers to what's next. Biotechs are coming forward more and more with perhaps minor modifications on existing chemistry, or modifying the absorption or other characteristics of a known and de-risked target, and coming to market and saying, well, a small bite or a small piece of a larger pie is worth our investments. And the VCs, the investment community, like I said, the ecosystem is really all participating in this. Each of them are enabling this herding that we're seeing. So yes, a Me Too or a Me 7 may not sound that attractive, but when you have a de-risk target and an ability to potentially launch something 2 or 3 years, say in 2025, 2026, versus going after a novel target and having that launch in 2030 or beyond, you have to make some hard kind of business decisions along the way, but there's an impact here. So beyond the concentration on targets, there are other pressures. I won't go into a lot of details, but it's worth pointing out that there's also not only a trend to go after the same targets, but there is this kind of industry chasing of modalities and fear of missing out, right? If cell therapies or protein degraders or ADCs or bispecifics become the next big thing, pretty much every company, every big pharma is going to want to have that technology in-house. So you're seeing the development and the investment in all of these different companies on these same modalities. And many of those modalities, to de-risk further, are going after de-risk targets. So targets that are already in the clinic or have already been validated, but now you're just bringing a new modality, a new way to attack that target. So this is having an impact on the way in which big companies in particular are making decisions about their pipeline. And we'll talk about how that's manifesting on the discovery side in the next few slides. But it's not only the targets, it's the modalities. You have these trends, and you have a risk of diluting resources, not only across targets, but also across modalities. And in addition to these elements, you have things like practical considerations. Again, there's probably too many for the purposes of this talk, but just to point out, this is a slide we actually usually use for biomarker-driven trials, and pointing out how challenging it can be to run a biomarker-driven trial, how to find those patients, make sure they're getting tested for the biomarker, making sure they enroll. But the scary truth is that the entire clinical trial engine is stressed to a level that it probably never has been before. It's more and more challenging to run trials. It's more competitive than ever, perhaps at least in part due to the herding and all of these trials for the same assets, all competing for the same patients. This engine cannot sustain all of these trials, at least not with the current patient recruitment paradigm that we have. You're needing a greater and greater proportion of patients within a given tumor type to actually enroll in trials. And historically, this has not been the case. You have competition for sites. You have high quality site staff leaving academics. You have COVID, which has had an impact. The cost of trials, opening trials, site activations for sites that never enroll a patient, and on and on. So the volume of trials cannot be sustained. And this brings you back to the question as to, are we making the best use of the clinical trial engine? Are we delivering the best value to these patients by providing the same many of the same targets over and over again? Lastly would be the economics, right? So if you think about the significant increase in herding, you have to ask, is there a rationale for this practice in terms of the financials? Does an investment in these Me Too's or fast followers generate substantial value, both for patients from a commercial perspective, as well as, again, for those patients that are receiving those medicines? Does this warrant biopharma R&D resources for such a practice? To characterize the commercial viability of late entrants, this is, I believe, another Nature reviews that looked at 29 classes of drugs after the year of 2010, over 100 different products in total, and looking at how much of an advantage did you really have by being first to market. And the analysis shows, and this has been shown elsewhere, that first-in-class oncology products have a significant advantage over followers, as long as they have kind of a medium therapeutic advantage, right? You have to have an active medicine. And not shown here is interesting is this advantage, this first mover advantage has actually grown substantially over the preceding decade. The market dynamics are challenging for late entrants that do not have a product profile that is very clearly differentiated, perhaps in multiple ways, and many do not. But there is still value that is being extracted by some of these novel medicines. If you're talking about a very large market, again, a small piece of that market is still potentially an incentive to go forward. So what about the earlier money? Where is this going into discovery versus into late stage assets? This slide for us is objective evidence that the field is recognizing that there is a paucity of novel targets. The increase in herding activity has already shifted the oncology deal-making landscape. You can see here on the left-hand side, the percentage of deals of in-licensing deals that large cap biopharma are making to bring in new assets or platforms. And what you see here is that deal making from left to right over the past decade plus, the percentage of deals that involve phase 3, phase 2, phase 1, or preclinical. And just 5 years ago, about half of the all oncology deal making was for clinical stage assets, so phase 1 through phase 3. Whereas the bulk of activity over the last couple of years, you can see particularly in 2022, has started to rise in terms of preclinical. So this is a fairly dramatic paradigm shift. We would argue is driven by the oversaturation of those same targets in biopharma pipelines and the need of these companies to differentiate from competition by unlocking truly novel drugs and first-in-class potential. It's still early in these trends, so certainly some caution here in terms of how we're interpreting this. These cycles tend to take a while to play out. Certainly when you're talking about a novel mechanism, you're looking at many, many years of preclinical through clinical development before that would get to market. And we certainly recognize that there's a decade prior of a very different balance in portfolios with more of those dollars going to late stage assets. So where and into what are these investments going? Getting to the second and third part of the talk would be they're going towards novel discovery capabilities, novel technologies, be it experimental, computational, multi-omics. Target discovery historically was dominated by traditional approaches, right? Screening, libraries of compounds, et cetera. There's been advances in science, advances in technology. Now we're in an age where techniques and multi-omic approaches really have exciting potential. More and more companies are looking at things like undruggable targets, which obviously is a misnomer. They're undruggable only because we didn't have the tools to drug them. We're breaking down many of those barriers. So combining these modalities, combining these omics, these different methodologies has great potential. And that really fits into the last slide of the first section is these next wave of therapeutics, these next wave of discovery methodologies that are going to get us there. They need to be focused and they need to be balanced, the way in which we're thinking about investing in our pipelines if we want to develop truly novel targets. The first part of drug discovery is obviously identifying a good target. We need biological rationale. We need to validate that target and we need to invest. We tend to over-index on the identification part as a community, but the identification component is only valid if you bring in your rigorous validation. You really need to prove that these targets are relevant. And having a balance between being enamored with new technology and deploying it thoughtfully is really key. So to optimally advance the field of target discovery and target ID, we would make the case that we should be combining wet lab, combining these exciting technologies, but also looking at patient tissue. So in silico, wet lab, actual patient tissue coming from patients with cancers rather than only modeling some of these things. Bring in the sophisticated in silico experimentation on large scale data sets is very compelling when applied with tissue, with some of the wet lab technologies that we have. So hopefully I've convinced those listening that there's a need to do more in the early target ID space. We want to be more innovative. And I'd like to pivot to tell you a little bit more about what we're doing at Caris just in our discovery unit, But to understand that, I'll briefly go over how we're going to make that happen and why that's differentiated. So what we're doing on the target ID side, we've thought about, how do we take advantage of what is really a scale of tissue and a scale of data that is unmatched and is enabled by our underlying business, which is profiling patients with late-stage cancer and providing that information back to a treating physician. So I have a few slides here to talk about Caris, and then I'll talk about how we're applying the Caris backbone to that target ID and drug discovery space in a way that hopefully is addressing the paucity of novel targets. So if you don't know much about Caris, we are the leading next generation AI tech bio company and precision medicine pioneer. We have been around for over 13 years, again, primarily focused at the point of care over the early parts of the life cycle of our company. Having a patient comes in, understanding that they're going to need a biopsy, having that biopsy sequence, we get that biopsy, we send the information back to the treating physician, and they can make an informed decision based on the genetic information and other information that they have about that patient. That is the backbone of who we are as a company at that point of care, extremely patient-centric. But over the last several years, we've spent more and more resources and more investments in helping biopharma advance their pipelines. And one of the ways in which we do that is through Caris Discovery, which we'll talk more about. The footprint that we have is, again, unmatched. We have 4 precision medicine laboratories between Texas and Arizona. On the left side and the bottom right of that figure in purple, you can see our R&D lab. We'll talk a lot about that. Almost 60,000 square feet for wet lab capabilities, where we have access to all of that tissue that's coming in for our sequencing volume. We have a footprint of over 50 NovaSeqs, which is the second and largest NovaSeq installment in the world and the largest sequencing power in the world. We apply all of that sequencing power to those biopsies that come in from treating physicians, from pathologists. Last year, we sequenced over 100,000 patients, so 100,000 different exomes and transcriptomes. The philosophy of Caris's approach to precision medicine is that comprehensiveness matters. And being truly comprehensive does not mean looking at a subset of genes, it means looking at all the genes. So 23,000 genes, whole exome, whole transcriptome, DNA and RNA. You can see here the numbers are pretty staggering, over 240,000 exomes, almost 300,000 transcriptomes. With that comes a huge repository, a growing repository over time of images, of patient tissue. We have over 10 million residual slides. We are digitizing the majority of those slides, which we started a couple of years ago. And then we're combining all of that data with clinical information from a variety of sources that I'll talk about in a moment. And when you do that, you create an incredibly powerful, matched clinical molecular data set that you can interrogate to help advance translational, clinical development questions, et cetera, as well as starting to apply machine learning and AI to advance kind of even more sophisticated signatures in the next wave of biomarkers. The backbone of our test is a best-in-class molecular blueprint where you're getting exome and transcriptome, DNA and RNA, full gene coverage, 23,000 plus genes, of course, all the different signatures that you can pull out of the DNA. On the RNA side, again, full coverage, lots of things you can do that are really exciting with expression here. Of course, you can call fusions better. And then we also do immunohistochemistry, which is really important for patient care at the point of diagnosis and treatment, but is also really important because all that information, all that IHC, which is relevant in the context of clinical care, is fed back into our database. So we have that information. What I've showed you in the last couple of slides was primarily our tissue-based sequencing. We're also doing this in blood. Again, going to be pretty disruptive to the field. We have launched this with a limited number of sites and with some of our biopharma partners, looking at whole exome and whole transcriptome in the blood. Pretty rare to be looking at RNA in the blood. It has lots of advantages, not only potentially to call fusions and others, but also to down the road look at expression potentially, but also to help inform the calling of DNA. Many liquid biopsies struggle because there's inadequate amounts of nucleic acid, of DNA. when you have the DNA and the RNA for Caris Assure, which is our liquid biopsy assay, you have a much greater ability to detect material and to make calls on these patients. We also sequence the buffy coat. This sequences the white blood cells, so you have germline. We also are able to account for CHIP directly, not through an algorithm, but actually directly remove CHIP, which is a important cause of false positives, and pretty innovative in terms of being bold and going after the most comprehensive assay we possibly can, get everything we possibly can out of these valuable specimens, not only in the form of tissue, but also in the form of liquid biopsies. And then putting all that best-in-class comprehensive molecular data and applying it to the patient journey as it relates to clinical data. And this is not our in-house expertise necessarily. We really excel in that molecular space. So we have chosen to partner very heavily to bring in clinical data sources from third parties and try to establish the most complete patient journey that we possibly can. So on the far left of the slide, at some point that patient record is going to begin, and at some point on the right-hand side, it's going to end. At Caris, we have access to some of that clinical information. So for example, we get progress notes that are submitted when we receive a biopsy before we do the sequencing. That is a window in time, perhaps 90 days of the window leading up to when that patient received the biopsy. But it doesn't tell us what happened afterwards, after that patient got treated, and it doesn't tell us what happened to that patient perhaps in prior lines of therapy if they had received any. There you need to look at things like claims, you need to look at things like EHR, depending on what the questions are that you want to answer, you will have a different need in terms of that clinical data. So we are trying again to piece the puzzle together with as many forms of that clinical data to complete that patient journey and attach that, link that to the molecular data so that we can use for development, for discovery, and other purposes. And this is what a multimodal kind of dream gold standard data set would look like, where you have deep, comprehensive molecular data, exome, transcriptome, you have tissue that you can do validation studies on, the H&Es, and then you have curated clinical data that can tell you about that molecular information, about that biology in the context of the clinical treatment paradigm. So this is what we do as our core business. We have been profiling patients for over 13 years. We've launched exome and transcriptome 3 or 4 years ago. So we have been amassing a large amount of data and a large amount of tissue. Caris Discovery is taking the first part of this presentation and saying there is a significant need, a paucity of targets in the oncology space, and we are sitting on an unmatched data set, an unmatched biobank. Let's interrogate that tissue and use that data to develop novel targets. And that's what we're doing. So most companies that tend to use tissue rely on kind of ad hoc through biopsies and other ways, other collaborations with academic partners, et cetera. We're fortunate because of the volume of patients that we see, over 100,000 last year, probably over 130,000 plus this year. That's tremendous growth. And that means we get a lot of tissue that we can use, and we can apply this to not only the data sets that we have, but a really talented laboratory R&D group that we have led by Heather O'Neill and Milan Radovich and others who are applying that data, applying that tissue to bring forward novel targets. And the way we like to describe this is that any one of these individual components that you see here in the honeycomb, the data, the tissue, the wet lab, alone, they may not be completely differentiated, or they may not be the only place in the world that these things are being done, but nowhere in the world are this many resources, differentiated resources coming together in one place, and nowhere in the world are they coming together at scale that we have at Caris. So fundamentally, to break down what are the differentiators that Caris has, and now I won't get into a really deep dive or technical talk here, but if you're thinking about how do we apply tissue, how do we apply data to developing a platform that will identify truly novel targets, We take the tissue, we have an aptamer-based approach that we use, which is what we like to call unbiased. I'll talk a little bit more about that on the next slide. We have a few other techniques, but I'll primarily talk today about the aptamer-based approach that we use patient tissue from patients with cancer. We then take that information and validate it on the bottom half of the slide from an in silico perspective and validate, characterize, back and forth, making sure that we're understanding the relevance of a particular target. Are we certain that there's an unmet need? If we were to bring this forward, if we were to partner with a biopharma company to develop a molecule against some of these targets. But these are the two biggest differentiators that we apply from a discovery perspective. So to double click on the tissue component. So we take an aptamer library, large, complex, and if you haven't worked with aptamers, they've been around for some time. I think the way we're using them here is a little bit unique, though, from what most individuals are used to. We are taking large, complex libraries of unenriched aptamers, which are simply single-stranded DNA. We mix them with tissue from patients with cancer. We can choose, a tumor type, colorectal cancer or lung cancer. We can choose a subtype of cancer, say, post PD-1 and non-small cell lung cancer. However, we want to approach this to identify targets in a population of unmet need. We put those aptamers with the tissue. We isolate the aptamers that bind not only to the tumor tissue, but we do isolate the tissue that is bound to the aptamers, that is the cancer tissue, and we remove the aptamers that are binding to normal adjacent tissue. We amplify those tumor specific aptamers. We have multiple rounds of enrichment, and then we purify and identify by mass spec those aptamers to find out, okay, we let the cancer tissue tell us where it's binding, what's important, what's specific to the cancer, and then we identify what those sequences are and understand what that underlying target is. We then, once we have a target of which we, for every one of these experiments, for these campaigns, we're going to have, let's say 50, 100, 150 or more targets. We then put it through a really rigorous set of validation experiments. Some of those are wet labs, some of those are in silico. You can see on the left-hand side, we start with a literature analysis, make sure that we understand the potential structure and function of this target. Do they have a known role in cancer? Where are they found? Is it on the cell surface? Is it intracellular? Is there a landscape of known literature out there? Are there patents on this target? Are other biopharmaceutical companies already interrogating this target? We go into gene expression profiling, understand the nature of that target between cancer and normal expression. We do have targets that range from cellular to intracellular targets. The flow here is really just for a hypothetical cell surface target. But you can see here, if that's the case, we would take that through a cell surface validation protocol. We do cell surface shaving, we do biotinylation, membrane fraction analyses. We then go to antibody development for targets that look good up until this point. This is probably the long pole in the tent, right? Developing antibodies or identifying commercially available antibodies to run your IHCs, your Western blotting, and so forth. We do cell-based assays, ICC on intact cells, flow, cytotoxicity assays. And then we go back to the tissue. And again, this is an advantage, right? Starting with the tissue, but also being able to go back to the tissue, do immunohistochemistry first on TMAs, and eventually get down to immunohistochemistry on whole slides from actual patients with cancer. And then we have the ability to not only look at the tissue itself, but we've sequenced all these patients. So we have whole exome and whole transcriptome, as well as clinical information on these patients to cross-validate and feel good about whether these targets are something that we think is viable and can be taken forward towards potential molecule development. So this is our kind of current pipeline as it relates to a variety of targets across different modalities. As we're characterizing these targets, we have certain characteristics that pop up that would make it more or less relevant for a given modality. Obviously, if it's on the cell surface, it's going to be something that's relevant for an ADC, a radio pharmaceutical, a bispecific, or cell therapy. If it's something intracellular, it's going to be, traditional small molecule potentially, or protein degradation, molecular glues, and you can start to divide up these targets and look at not only what are the characteristics that would make them optimal for a given modality, but what are the tumor types that these are expressed in, and how do we want to further validate those across tumor lines or across some of the patient tissue that we have. So we take it through the wet lab validation. We're happy to report, we won't go into the individual targets today, and obviously they're de-identified for the most part. But if you look at some of the targets that have popped up, these are well-known targets, things like TROP2 and others that have been taken forward into development by companies and onto the market. Obviously, we're not going to pursue those targets. Our mandate is to look for novel targets. Nevertheless, it's comforting to see that known targets are coming up through this identification and validation protocol. We are not a big pharma. We are not a drug development company. So Caris leverages its differentiators in that tissue, in the data, in the wet lab to bring targets forward and validate them. But at some point, we need to get those targets in the hands of companies that really excel at creating molecules, going through the preclinical work, and eventually getting those targets and molecules to patients in need. Our primary way of partnering with biopharmaceutical companies is through a target out-license model, which is a little bit more transactional. We provide validation packages to companies to then take forward and add to their pipeline, add to their portfolio. We also do joint discovery work. Because we have the tissue and the data, it's nice to have a collaboration where not only are targets being provided and the options provided to those targets to some of these biopharma companies, but also to continue to work with them to leverage, the best of their validation pipeline and the best of our validation pipelines, be it tissue, data, et cetera. And we do some work on the far right in terms of targets that are not identified by Caris, but are coming in from the external partners pipeline as well. We have a few proof of concept deals that we're very proud of. A couple of those, two of those have been with Xencor. On the right-hand side of the slide, you can see an announcement we had earlier this year that was building on an agreement we actually had in August of the prior year in 2022, essentially out-licensing targets, getting those targets and those validation packages in the hands of a company that is well known to be able to bring forward medicines and to do it rapidly, right? Some of those targets will fail. We want them to fail fast. And the targets that have potential, we want them to get tested and hopefully bring those, again, to patients with unmet need. We announced our Caris Discovery platform just earlier this year. We've been working on it for several years, but kind of have just come out at kind of a critical mass of having enough targets, having enough different validation protocols in place where we feel really good about these validation packages. And this will not be the end. Hopefully before the end of the year here, we'll have a couple more opportunities to announce some deals with big pharma and with other companies. We are generating targets and we want them to get to patients in need. We can't do that alone, so we will continue to partner with the biopharmaceutical industry. So I believe that ends the presentation, and I'll just kind of conclude by saying, hopefully I've convinced you that there's a paucity of novel targets. We need to do more as an industry. We need to do more to balance our pipelines and balance our investments. There appears to be a trend of this in biopharma with some of the recent deal making coming into kind of more and more investments in early platforms. I think we also need to think about what can we do that's truly differentiated and avoid having multiple different omics, multiple different platforms being applied to drug discovery and target ID in isolation. We need to combine these capabilities, bringing together wet lab, bringing together things like patient tissue, and bringing together data at an unprecedented scale and technology to help find new weaknesses in tumor cells that we can go in and exploit. Most patients do not respond to most therapies, and that is a problem. We need to address this, and we won't be able to address it overnight by only investing in targets where we've seen this hurting occur. So let's have a call to action. Let's have a call to arms here to collectively invest in early target ID and really think about how we can come up with truly novel targets. So really appreciate your time and hope that there's some questions, and I'll open it up for the Q&A now. Thank you.
Thank you very much, Dr. Lamon. I am aware of the time we have left, so let's jump straight in with the questions raised. Our first question addresses the limitations of traditional target discovery approaches. Could you please share some insights on this topic?
Sure. I think one of the last things I said there in terms of isolation is a big one. Most people are good at one thing or a particular area, whether it's a technology or science, scientific discipline. I think the challenge to really achieve those transformative leaps forward and really have an opportunity to generate truly novel targets in ways that we would have never been possible, you have to be good at a number of different things. And I mentioned it in terms of multi-omic approaches. It's not that simple, right? Each of us are good at certain things. As I mentioned, certain companies excel in certain elements of drug discovery or certain technologies. Capital is a real problem too. The ability to bring in multiple technologies and do this multimodal type early discovery work is not going to be cheap. So most companies won't be able to do it alone. At Caris, we're in a fortunate position because we have a core business that pays the bills and generates a lot of the differentiated value that we're able to then use towards discovery, right? The tissue and the patient tissue and the data that I spoke a lot about. But that's not the case for everyone in the community. And if you're talking about bigger companies, there's a whole different set of challenges, I think, in terms of the discovery approaches there, right? How is the reporting structure? What are the motivations for that particular company? What are the revenue gaps that exist, and what do they need to fill, and the timing there? So it's complicated, but I think coming together as much as we can in terms of collaboration and building combined multimodal early drug discovery approaches are going to win the day.
Thank you. Our next question is actually an intriguing one. Are laetrile as well as cannabinoids being targeted as potential therapeutics for cancer therapy? If not, then why?
So I don't believe either of those are either cannabinoids or laetrile, if that's how it's pronounced, is FDA approved. I believe the former, the cannabinoids, have been used in terms of management of pain and other issues associated with that patients with cancer endure, either inherently or as a result of chemotherapy and other therapeutics. Not an expert in this area. However, the opportunities to explore biology are many. So the same vein of what I just mentioned in terms of bringing together technologies and different companies, I think we need to look at novel ways to not only discover new targets, but think about combination therapies. So whether that's with cannabinoids or others, we have to bring different disciplines of biology and different pathways and scientific expertise together, because the immune system's really impressive and really smart. Human physiology is really impressive and adapts and has compensatory pathways. So, that kind of silver bullet is usually not going to exist. And many of the driver mutations that we've had significant advances with, I should say, over the last couple of decades, those low-hanging fruit are going to go to the wayside, and we're going to have to be more sophisticated in terms of how we select patients and in terms of how we go after them through biology, therapeutics, and diagnostics.
Thank you. Our next question explores the integration of wet lab and in silico capabilities for target discovery purposes within biopharma companies. Could you share insights into how these two facets are combined and synergized to enhance the target discovery process?
Sure. So I think in silico is something that has, AI and machine learning has gotten a lot of buzz, and rightfully so, over the last, many years. In particular, the last 3 to 5 years, we've seen a number of companies emerge in this space that are doing almost pure in silico efforts in terms of identifying novel targets. And it's incredible what can be done. I mean, I'm not a deep expert in this area, but I mean, look at things like ChatGPT and others. Imagine what that could do for some of the things that the human brain alone, or even kind of standard computing, is not going to be able to uncover trends or novel ways to approach targets. So I think the in silico machine learning and AI is really important. I also really think, and hopefully this was evident throughout the presentation, that patient tissue and getting the right models to really study some of these things is really important. I think either one of these in isolation is problematic. So I think having more and more access to actual patient tissue and interrogating that tissue in a number of different ways, right? We're doing this in a few different ways at Caris to identify novel targets, but there's other approaches as well that can help us understand, what are some of the unique aspects of these tumors that can make them targetable through novel therapeutics. And there's the kind of lower hanging fruit would be, something on a cell surface where all we really need to know is that this is a way which we can identify and attack the tumor and not destroy the surrounding normal tissue or, other organs. The other is understanding if it's an intracellular target or something that's related to the abnormal proliferation, the abnormal metastases. That is more challenging because you're going to have to show cause and effect. So again, more and more technologies and the right models are going to be needed. So I think it's a blend of both the in silico and tissue that is really critical.
Thank you so much, Dr. Lamon. Unfortunately, that's all we have time for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. As with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thanks again, Dr. Brian Lamon, and we'll be back soon with the next talk.